All right, so now Bob is going to add 500 microlitres of 70% ethanol into each of the 2 new tubes that he has prepared.
So now just take 500 microlitres of 70% ethanol
into 2 clean 1.5 mil microcentrifuge tubes.
And now a transfer.
Now you're going to very carefully remove 400 microlitres of supernatant from these tubes that have been centrifuged, taking care not to disturb the palate, and add that supernatant. Add that supernatant to our tubes that contain the 70% ethanol. And in the transfer of that supernatant, we avoid transferring any of the solid plant material into the ethanol. All right, now carefully pipette up and down to thoroughly mix that lysate and ethanol.
Important to note that I'm using a new tip every time I go from one solution to the other. That's to make sure or reduce the likelihood of prevent any cross-contamination. Okay, so next step is?
All right, in the next step, we're going to take 2 of the mini DNA extraction columns. Here they are. These are provided in the kit. There you are, Bob.
Thank you, Barry.
All right, and you are now going to transfer 800 microlitres of that cleared lysate and ethanol mixture to each of those 2 columns.
How much? 800?
800 microlitres.
Yep.
Of your cleared lysate ethanol mix.
And then?
So this step will actually bind the DNA to the resin that's found in that purple column.
Okay, and then we'll take that.
Yep.
That's microcentrifuge tube.
And we'll centrifuge for 1 minute at full speed at room temperature.
Okay, so let's go to the microcentrifuge. And of course I'm going to go and do this for wet plant number 2. And once I've done that, remember it goes across the axis of the rotor, and then we start the centrifuge step.
Thank you, Bob.